Okay, today we're going to talk about growing bacteria in liquid culture. So we're going to look at basically how a cell population increases over time. And so we need to know a few things. Well, how do bacteria grow? How is growth affected by different environmental factors? How do you interpret growth curves that we're going to generate as we measure these, the growth? And how do we measure it? How is it monitored? And things to just take note of is we're going to need some serial dilutions. And what do serial dilutions do? What are they? We'll talk about that. And I'll give a little notes at the end about how the experiment is done. Okay, so bacterial cells grow by binary fission. Basically, you start out with one cell, which is called the mother cell. And you end up with two identical daughter cells at the end of one of these cycles. Now, what's going on here? So, in the mother cell starts to prepare to create two more cells. It's going to replicate its DNA. So we can see the replicated DNA right here. Makes more cytoplasm, cell wall, and cell membrane. And so basically you can see right here, it looks like there's about 2 volumes worth of cells. And the original chromosome is attached to the cell wall. Now, as more of the stuff, right, the cytoplasm, cell membrane, and cell wall gets made, this starts to pull the original chromosome, in this case, to the left. The newly replicated chromosome will also become attached to the cell wall, and that's going to get pulled to the right. And so you can see that the chromosomes move to 2 different cells, which you can see right here. And then you get the septum forming in between, which will pinch off and make 2 cells. So at the end, right, we have 2 identical daughter cells coming from this original mother cell. Now, we can monitor this by various different ways, but cells in liquid grow in a predictable manner. So usually when you see cell growth, you will see a growth curve that looks something like this. And there are certain hallmarks of this growth. Now, first of all, we're basically doing a time course, so we're looking at growth over time, so a point where we start here, as far as we tend to read. And then on the y-axis here, we have number of cells. Now, there are multiple ways to measure the number of cells, and I'll talk about that in a few slides. Okay, so looking at the growth curve. So I said it's kind of predictable at how cells will grow in liquid. And so what we can see is we can break up into lag phase, exponential or log phase, stationary phase, and decline or death phase. So what's going on here? So lag phase is usually when you first add cells to liquid media. They need to adjust. They need to make enzymes. They need to grow and prepare to start growing. So they don't grow very much for a while. At some point, they get all those things. And they get those things, and now they can start growing more quickly. So they start doubling. Right? So they start entering this point where now binary fission is in full swing, and the culture is basically doubling over a certain amount of time. Okay, so what happens? They go really well in this exponential phase, and then they get to a point where they start to tail off. What's going on here? So this is start to enter stationary phase. Now, at this point, the culture, many of the nutrients are depleted. There's increased in waste products. So the rate of growth is sort of balanced by cells dying. Right? So it sort of flattens out again, similar to what we saw in lag phase. And this, we enter what's called stationary phase. And eventually, the conditions are suboptimal and start getting the culture begins to die. And this can take actually a very, very long time. Cells can remain in stationary phase for an exceedingly long time. Okay, so I mentioned this thing about doubling time. What do I actually mean by that? So all I mean is how long does it take a culture for its number of cells to increase twofold, to double? Now, we measure this in exponential phase, and this is a characteristic of how the cells are growing. Now, different cells, different bacteria grow at different rates. They will have different doubling times naturally. Then there can be different environmental factors which may cause them to speed up or slow down. And you can easily look at the slope to get an idea. So here, a steeper slope during exponential phase means that there's a shorter doubling time. So if there's a shorter doubling time, the cells are actually growing faster. A longer doubling time means that they are growing much more slowly. And so, like I said, there are a lot of factors that can affect this. There are factors such as what nutrients are there, what temperature, bacteria like different temperatures, how much oxygen, pH, how much osmotic pressure, and a host of other things that are not listed here. So there's lots of environmental factors. Okay, so I'm going to talk about interpreting these graphs uh, uh, using this experiment from this paper uh, where they watched the growth of Bacillus flexus. And you can see um, so some of those cells here. They look like typical bacilli. And they added different amounts of sodium chloride into the media, and they grew them separately in these sodium chloride varied media. And asked, well, how do they grow? They monitored them over time, and the way that they monitored them is they looked at absorbance at 600 nanometers. Now that's a common way to do this. We'll talk about what that means in a little bit. It's one of many ways that you could do this, but this is a very common way to do this. And so they just measured, how many cells they have by measuring the absorbance over this period of time. Okay, so how do we interpret these graphs, and what did they tell us? And they can tell us a lot. And the easiest way that I like to do this is I like to systematically go through the separate curves and compare them to each other. And the other thing I should mention is we don't just compare the curves, we can compare distinct parts of the curve. So let's look at this first curve, which is 3.5% sodium chloride in the media, compared to when there's 7% sodium chloride in the media. Okay, so if we look at this, there are a few things going on. First, if we look at lag phase seems to be increased, like the time that it takes to enter into exponential phase, so that the time that the bacteria spends in lag phase is increased when the salt concentration is higher. So it compares the 7% to the 3.5%. Okay, but the slope, if we look at the slope, once they get into the log phase, the exponential phase, they seem to have similar slopes. So they seem to be growing at the same rate. So what does that tell us? So here, the cells need to adjust to the higher salt. Well, at 7%, it takes them longer to adjust to that. But once they do adjust to it, they have everything they need to grow just fine, and they grow just as well as when there's a lower amount of salt. And they reach a similar level where they start entering the stationary phase. It takes a little bit longer for the 7% to get there, but they eventually reach the same point. Okay. Now we compare the 7% to the 10%, and there are some drastic differences that we can see here. First is that the lag phase is yet increased more. So it takes even longer for the cells to adjust to the higher salt concentration, in this case 10%. Second thing is that when they do start growing, when they enter the log phase, well, now the slope of this curve is less steep than it is over here. So they're not growing as well. They're growing more slowly. It's taking them a bit longer to divide. And they eventually end up reaching the same level. They haven't reached it quite yet, but they should get there if we were to continue this experiment longer. So the conclusion is that there is a longer doubling time occurring in the 10% culture, 10% sodium chloride culture, and a longer lag phase. Now, what's happening down here? If we look at the 14% sodium chloride culture, well, it's just not growing. The cell can't handle that much sodium chloride, and it's just sort of staying level and not growing at all. Okay, so here we look at another factor for growth, another environmental factor that's not often considered. Now, we grow a lot of cells, and you've already done in class where we put them in their shaking. Now, a lot of bacteria need oxygen, and the shaking basically helps to mix oxygen into the media. Some bacteria don't need a lot of oxygen. Shaking them doesn't help. But things like E. coli and many of the bacteria that we use, shaking helps them grow better. And so here we have an orbital shaker. We have a couple bigger ones in the lab, and we have our media in some sort of container. And this little platform down here rotates, and it will cause the liquid to slosh up on the sides and go back down. And all that's doing is mixing the oxygen into the media. If you look at E. coli, so here's a graph for E. coli where we're measuring the optical density or the absorbance at 600 nanometers over time. And you can see if we don't shake it, they grow. They don't grow very well, but as we increase the speed which we shake them, they start to grow much better. So they get into steeper log phase. They reach higher levels to stationary phase. So this aeration is a very important thing. Okay, so talk about measuring um, the, the, the amounts of cells in some way. And so there are multiple ways to do this. Now, there are direct methods. There is the direct microscopic count, which we will not be doing. We'll talk about a little bit. There are plate colony counts, which we will be doing. There are indirect methods also. We can measure the absorbance, which is what I just showed, the experiment I just showed you. I'll explain in more detail next couple slides how that works. You can also measure metabolic activity. And so if there's a particular enzyme that the cell makes, sometimes the cell, you get more cells, there'll be more enzyme. You can track that enzymatic activity. So that's one way of following the growth. And you can simply just take samples of the cells, put them in centrifuge, spin them down, dry them out, and ask, well, what's the mass of the cells? Now, all of these are what give us this y-axis. So you can pick any one of these and they will work. There are some benefits to some of them and drawbacks to them, and you use whatever works best for your experiment. So the easiest way to do this is through spectrophotometry. So you measure this absorbance, also known as turbidity, or basically the cloudiness of a culture, right? And this is used very commonly. We saw the previous example. It was used in that. And this is just an easy way to sort of measure the growth. And basically looking for how cloudy a sample is. So first thing we do is we have to sort of get a baseline, and we do what's called blanking our spectrophotometer. So here we have a spectrophotometer. There's a light source. We have our sample, and light will pass through the sample and get read by the spectrophotometer. So we have to say, well, if there's nothing growing, what does it look like? And so we take whatever media that we're growing our bacteria in, no cells, and say, well, what does that look? We set that to 0 absorbance. Because what's going to happen is as the light passes through here, if there are cells in here, they're going to absorb some of the light. And that's what we see here. So here, the difference here now is that we have a culture that's grown. There are cells in the culture, and the light source, the same amount of light is entering our sample, but not as much as getting through. And this we measure how much of the light is absorbed. And so you can see here a culture that is very cloudy. You have done this using a nanodrop. It's one of our spectrophotometers. We have other kinds of spectrophotometers also. Now, the biggest advantage of this is it's just very easy to do. But there are some drawbacks. And so it depends on what you're doing, whether or not those drawbacks matter to you. So what it can't do is it can't distinguish if there are multiple bacteria in your culture. So if you want to see how one bacteria is growing, you won't know if it's contaminated. That will just become more cloudy. You won't be able to tell how much of the bacteria you're interested in versus a contamination is growing. If you have 2 bacteria that you want to grow, well, you're not going to know the difference. They're both just going to make things cloudy. The other thing that can happen is sometimes bacteria will produce some sort of byproduct, and it might cause the culture to become cloudy, not due to more cells, but just due to the presence of whatever that metabolite might be. The other thing that you do, so for E. coli, this has been worked out for decades. But anytime you work with a new organism, you sort of need to make a standard curve so that you know an absorbance value of whatever number. How does that correlate to the number of cells that are actually in your sample? Now, a way of directly measuring microbial growth is using a direct microscopic cell count. And so basically what you're going to do is you're going to count all the cells. You're going to look at them under a microscope and count, well, how many cells are in that sample. Now this can be automated, which would be fantastic. If it's manual, that's a bit more work. But you need a certain tool before you even do it, a thing called a hemocytometer. Now, what this is a little chamber, and you add your culture to this. When you look at it under a microscope, you will see a grid. The grid corresponds to a certain, each one of these squares corresponds to a certain volume, excuse me. So it corresponds to a certain volume. And so you count the number of bacteria. So I'm going to count everything on that I can see, or I'm going to count in 10 or 100 squares. Well, you get your count of your bacteria, and then you know your volume, so you get number of bacteria per unit volume, milliliters, microliters, whatever it may be. Now, as I mentioned, we don't do this. We're not going to do this, but this is a very common technique in cell biology. So you have to use a microscope to do this. And as we're going to talk about serum solutions in a moment. We're going to use them for a different purpose, but you will most likely have to dilute your culture down a little bit serially to get individual cells that you can easily count. Now, good thing is you can see individual bacteria. If there are more than a type, if they look different, you can see them. You can count exactly how many are of each type. And you'll also be able to see if cells are alive or dead. All cells will be there. Method that we're going to use is we're going to use a direct measurement for looking at microbial growth, and we're going to do plates and what's called viable cell counts. And so basically we're going to look at how many bacteria are in a culture by taking some of that culture and putting it on an agar plate, letting it grow and seeing the colonies, counting the colonies that are there, and then multiplying back by any dilution factors we may have to figure out, well, what was the concentration of bacteria in the original culture. Okay, and so in order to do this, if you take whatever you're growing and you just put it directly on a plate, so we have this culture here, chances are you're going to get what's called a lawn. There are going to be so many cells that are going to all go together, you won't be able to distinguish any individual ones. So usually what you have to do is you have to dilute it multiple times. The most accurate way is to do a serial dilution. You take a little bit out of this one, you mix it with some sort of buffer solution in here, that dilutes it. Take a little bit out of this one, mix it again with another one, dilutes it even more, and you just keep repeating until eventually you see individual colonies. Now, well, how do we use these individual colonies to determine what the concentration was? Well, it depends. You have to think about what a colony represents. A colony represents where one cell has landed on an agar plate and it's grown through this binary fission, but in on a surface, well, all of a sudden you see a mass of cells. So that mass of cells, which is called a colony, represents where one bacteria has landed. So we can count how many colonies, and that's going to tell us how many bacteria were in our sample that we plated. And then we have a whole bunch of dilutions here. We'll have dilution factors. We have to multiply by those dilution factors and figure out what our original culture, or what the concentration of bacteria was in that culture. Now, this uh, method only gives living cells. Cells have to be able to form a colony. If they cannot form a colony, we can't see them. But the good thing is, many bacteria, when they form colonies, the colonies will look different for different bacteria. Not always, and we've seen this in class already. But if you can tell the difference between the bacteria based on their colony morphology, well, you can determine how many cells of each type were in the original culture. Okay, so what are we going to do? So what I'm going to describe here is we're going to do for each time point. So you're going to have to do this multiple times. And what you're going to do is you're going to take a small sample out of your culture at a particular time. This is going to be our concentrated sample. And we're going to determine how many cells per milliliter are in that sample. And so what we're going to do, we need to dilute it. So we're going to dilute it in a systematic way. So we're going to take 100 microliters out of our sample and mix it with 900 microliters of our dilution buffer. This tube, everything is 1/10 as concentrated, so this is a 10-fold dilution. Now we can repeat this many times. Each time we do that, we're going to dilute it a further 10-fold. So we can see here that we'll dilute, this is 10-fold, 100-fold, 1,000, 10,000, etc. And we're going to dilute our sample to, we have our dilution factors. This is very systematic, very easy to determine what we've done. Now we're going to take some of those when we get to the end of doing our dilutions, and we're going to put some on agar plates. Now, these first three are not very useful to us because they're so concentrated that we will not be able to see any individual colonies. We're going to plate these last four. And actually, this 10 to the fourth is also going to be very concentrated. but sometimes when cells don't grow very well, it's good to have that on your plate. And so we're going to put 10 microliters out of each one of these samples into a spot on our plate. So this is an example of a plate that a former student has done. And you can see we have our dilutions, 10 to the minus 4, 5, minus 6, minus 7. And this is done in 4 times. We'll talk about that in a moment. But these all represent 10 microliters. Each spot is 10 microliters, in this case, coming from this minus 4 dilution tube. And we'll have 4 of those, then 4 from the minus 5, 4 from the minus 6, 4 from the minus 7. And you can see here that you start with a very concentrated high number of cells, and they get less and less as we go from left to right. Now, there's a couple ways of thinking about this. I'll explain just the way that I sort of grew up learning how to do this. Is that the units that we're going to eventually express this in are things called CFU per mil. CFU represents colony forming units, and instead of calling them colonies, the term that's more appropriate is colony forming units. But the important part is that we represent them in the volume of 1 milliliter. Now we are only plating 10 microliters. So the number that we get here, whatever we count up, is for 10, in 10 microliters. We're going to want to express it in milliliters. So 10 microliters is actually 1/100th of a milliliter. So this is an extra 100-fold dilution beyond the dilutions that we did here. Now, if this is confusing to you, you don't have to think about it this way. We have a formula that takes all this into account. Like I said, this is a way that I was taught when I learned it, and for me it makes sense. It doesn't make sense for everybody. Okay. All right. So how do we, calculate the original cell density in that sample? And so here we have our, sort of layout for our spot plating. We have 10 microliter spots, 4 of them from the 10 to the minus 4, 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7. And we can see that, this is the example that we showed of what this looks like. So you'll be doing one of these for every single hourly time point. So you can see here's just the examples labeled as T0. So that T0 would be the 0 time point when you first start your cultures growing. This would be the 3rd hour time point. So you'll need 1 for each hourly time point. And so how do we use this this data? How do we convert it into numbers that are useful? And so we need to find out our original cell density, or OCD. And again, the units are in CFU per milliliters. So we need to get the number of colonies. We get those from counting spots on this plate. We need our dilution factor, which is which we've already determined, and the volume plated in milliliters. And each one of these spots represents 10 microliters, which is 0.01 milliliters. Okay, so let's have an example here. So if you counted 12 colonies total in the 10 to the minus 6 spots, and I went through and counted them, I hope I'm correct. I counted 12 colonies, and in all the 10 to the minus 6 spots. So we know the number of colonies. We know the dilution factor is 10 to the minus 6. What's our volume? So each spot, again, is 10 microliters. So this is 40 microliters, or 0.04 milliliters. We now have all the variables determined. We can determine what the original cell density was. We have our 12 colonies. We have our 10 to the minus 6 dilution factor multiplied by our volume, which is 0.04 milliliters, and we get 3 times 10 to the 8th colony form units per milliliter. Now we can take this, and when we get our colony counts, our original cell densities, we can compare those to the optical densities that we determined, our absorbance values, and see how they relate to each other. And we can get a standard curve. And so here we just have each of the time points. Well, what was the absorbance value, the OD600? How many CFUs per mil did we get for each one of those? And you just plot them, and you get a trend line, and then you can get the equation from this trend line, so you can determine, say you measure an optical density of 0.4. You follow that over to where it crosses the line, and you will get this many coliforming units per milliliter. Now, alternatively, you can take this 0.4 and you can plug it into the equation that you get for this trend line, and you can get a more accurate count. Okay, so what are we going to do this week? How do we do this experiment? Well, we're going to grow one bacterium in LB broth, and we're going to add sugars to it, either glucose, sucrose, or both, and at different temperatures. And then we'll compile this data into a single graph for you to analyze. So basically, any experiment you do, and this is also if you need to do your independent project, you have a similar kind of experiment, is you can take what media you use, to split up by the temperatures that you use, and in this case we have whether or not there's sugar. So this first case where there are no sugars, then we have just glucose, just sucrose, or both, and we repeat that at the different temperature. Now, this is just to give you an idea of things that you can do. We will, each year we will have some variation of this depending on how many students we have and how many of the different conditions we can go through. But if you're doing an experiment like this in your project, you may want to vary the media. That might be more important, and keep the temperature the same. You may have different sugars or salts or something else to add. And so you can generate a grid like this so that you cover all the cases for the variables that you are concerned with. And you can systematically go through them and generate growth curves for them and interpret them and hopefully get some good information. Now, before class, what you're going to need to do is you're going to need to do some math about how to prep media. And I'm not going to go through all of that here, but other than just to sort of tell you what we have. So we're going to use LB broth. We're going to use sugars, glucose, and sucrose. And we have concentrated stocks of LB broth, where we have LB broth, which is twice as concentrated as it needs to be. And sugar solutions are both at 40% weight per volume, glucose or sucrose. And so you'll need to find out, calculate how much you need uh, for making LB that has concentration of 1x, so the general working concentration for LB. And the final concentrations for the sugars should be 0.4%. And my favorite formula from chemistry, thing that I use the most, is right here. This is what you will need. And so you need to sort of figure out these calculations before you come to class. Now, definitely want to look over the protocol, read it carefully, because this experiment is not hard, but it takes some sort of prep to really get it going smoothly. So you're going to grow your cultures, and we're going to do small volumes. It's all written out nicely in the lab manual. You're going to take portions out at specific times. You're going to read the absorbance for them each time. At every hourly time point, you're going to serially dilute them and spot plate them onto agar plates for colony counts. So every half hour, you will take an absorbance value. Every hour, you would take a sample to dilute and to plate. And then from that, you will be able to determine a doubling time for each condition and compare them between conditions. Okay, well, good luck on the experiment, and we'll see you in class.